Hello everyone. In the molecular basis of inheritance, we should have little idea before we proceed with the replication. So the idea which we should have is that DNA replication is actually a biological process which produces two identical replicas of DNA. Suppose a DNA is, since I have made it in red color, so think that both of them are complementary. Both the strands are complementary to each other. Whenever we want to make a replica of each of the strands, suppose this is one strand, I wanted to write one, and this is the second strand. If I want to make a replica of one strand, I'll just open it. Second one also I'll open. And after opening it, I will use certain other nucleotides in order to just make the replica of what this strand had here and the similar replica at this place. This is our intention. We can do it very easily as we are aware that A will always pair with T and T will always pair with A. Similarly, G will always pair with C and C will always pair with G. With this idea keeping in mind, we have to make new strands. Now, one is completely red colored strand, whereas another one is a green strand. The green strand shows that there is a new generation or a new strand development taking place. And since this was the parental strand, now the strands which are formed new, they are the new strands. These new strands are semi-conservative in nature, in the sense they conserve. They have 50% parental character and 50% new strand. So one strand is parental, another strand is new. Now, when these two strands also want to replicate, at that time, the red one will again extend itself like a red. I'm sorry, extend itself like a red, whereas the green one will extend itself like a green. Now, this red one, which has extended itself, its complementary part will definitely be green because that is the way we are doing. And again, the green part which we have, extracted out, this green part will again form some other new type of or a new strand. So by this, the little variations occur and every time a new strand is formed. But again, the nucleotide content is still the same. So that is about, first thing is about the replication, that identical copy of the DNA will be made. Second thing is about the semi-conservative nature of the DNA, that it is partially, or you can say 50% similar to its parent. There is a region which is known as ORI, which is origin of replication region. And at this region, the strand, it starts differentiating, or you can say this is a site at which the replication process is initiated. The synthesis of the strand always takes place in 5' to 3' direction because these 5' position and 3' position has free OH groups, and hydroxyl groups initiate or help in the process of division. Cell division will occur, or not only cell division, I should rather say that addition of nucleotides can occur, or any biomolecules addition can occur only at 5' and 3' position of the glucose, but here it is a deoxyribose glucose. RNA primers are required in order to initiate the replication. So there is a tendency, or there is a requirement of RNA primer. So RNA primers are, suppose, 5 or 6 number of nucleotides are arranged, and that small area or small segment of RNA is taken in order to initiate the DNA polymerase activity. So this is the basic idea you should have before replication. Now, why does this process of replication occur? It occurs in order to preserve the genetic integrity of the species, and it also preserves the genetic constitution of an organism by undergoing very less change or maximum level of accuracy while the replication or duplication of the genome is taking place. In the eukaryotic cells, this process takes place in the S phase of the cell cycle, because you all have studied also that during the S phase, duplication of DNA occurs, and duplication of DNA means that replication is basically taking place. the rate of eukaryotic replication is much slower, and because of that, very high accuracy is maintained. So high level of accuracy is there in the strands which are formed. This also helps in transmission of genetic information from parents to offspring. That is it. Half of the parental strand is retained, so it is definitely forwarding the parental generation or parental information to the next generation. Replication of DNA was stated or was informed by Watson and Crick. So Crick, when he made the Watson and Crick, when they made the double helical structure of DNA or announced the structure of DNA, they did talk about the replication of DNA as well during that time. They said that these two strands they can be separated, and when they are separated, they can make complementary strands of their type. And each of the parental strand will act like a template, and it will allow formation of a new complementary strand. After each replication, each DNA molecule would have one parental and one newly synthesized strand. This is what we have discussed, that if we are starting with suppose red colored strands and we want to make replicate it, then the next will be some other color. And as we want to replicate the another these two as well, then again you have to think that one will be parental and another, I'm so sorry, this one will be orange, and the another one which is going to get added will be maybe of some other color, but here it will again be orange colored, because parent one always forms the new strand as orange one. So this leads towards the semi-conservative nature of the DNA. There was an experiment done in order to prove the semi-conservative nature of DNA, and it is there for understanding the semi-conservative nature of DNA little better. For this experiment, we need few substances. First thing is a E. coli. E. coli is a bacterium, you are aware, and this bacterium we need two mediums. One is, I'm so sorry, one is having a heavy isotope of NH4, like N15, and another one has NH14. Two mediums we need, two petri dishes which has, one has heavy metal, that is N15, and another one has N14. Why N? Because N is an integral part for nitrogenous bases. They need presence of N, so it will get incorporated in the DNA structure. This N will get incorporated, so we can see radioactivity being present. We need cesium chloride because we want density gradient separation. Yes, we need density gradient separation, and for that we need cesium chloride to facilitate the process. Next is the centrifuge is required. It is a machine in which the if we put some test tube and close it with the lid, then after it can be rotated at a very high speed. When something is rotated at a very high speed, there is a chance of separation of things of different density. So heavy metal will be present in lower proximity or will be present at much lower range as compared to the lighter one. Suppose this is a structure or a test tube if I have tried to make, and if N15 is formed, it will be formed much higher than N14. Suppose this is the N14. Why this is N14? Because it is light. And why this is N15? Because it is heavy. This is N15. So with this understanding, let us proceed further. So this is what is going to take place. We will take parental DNA. Now here one thing is important. You can see this first type. This is an example of conservative type of DNA replication. But the one which occurs or which we are trying to judge right now is this semi-conservative nature of DNA. You can see the blue one is a parental strand. So this is parental strand. Blue is a parental strand. But yellow is recombined, or you can say new strand which is formed. So again, if this one opens up, then blue one will always pair up with yellow, whereas yellow will have the yellow types only. And As an end product, what we see is we have 50% parental type and 50% of new type. These two are new and two are parental. So this is what we have to prove through this entire experiment. So we can see, at first, what we have done is we have taken this experiment is known as Messelson and Stahl experiment, and it was done in order to figure out that which that the density gradient separation. With the help of that, we can find out that semi-conservative nature. The DNA has semi-conservative nature. So, what did we observe during this experiment was, in the first generation, when we just initiated with the experiment, we saw that the DNA isolated from the cells were kept in an experimental setup, and they were switched to N15 NH4Cl produced. And how was this heavy metal produced? We are basically interested in putting the entire parental DNA, which contains N15. And how do we make it? We just put, like, take a petri dish and fill it with N15 kind NH4Cl. This kind of substrate is there, and DNA is allowed to be here for a long period of time and is allowed to replicate. So all the DNA which is formed will have this, or will have nucleotides having N15, which is a heavy metal. Now, after that, when we initiate the process of replication in the N14 medium, so now the medium will be changed. Once we take the DNA from which is already labeled with N15, then after, when in G0 or the first generation we are taking the substance, what we are doing is we are using a petri dish, and in this petri dish there is N14. We just want to judge that how much time is it taking, or how is the semi-conservative nature of DNA being proved. We have already taken N15. Now, if we allow it to grow in N14 and want the DNA to replicate, then the nucleotides which will be utilized will be N14. So that is what takes place. In the next 20 minutes, we see that DNA isolated after first generation or first round of replication, what does it produces? It produces equal amount of, or this band was higher intermediate in density between the high NH4Cl, heavy isotope of DNA, and light isotope of DNA. So in between something is formed, and this intermediate band made Stahl and Messelson understand that DNA molecules made in first round of replication was a hybrid of light and heavy DNA. This results fits the dispersive and semi-conservative models of DNA. But till now, it is not very well proved that it is a conservative model. So, in order to prove that, they predicted two distinct bands in the generation, an heavy original molecule and a band that had light newly synthesized molecule. Why? Because we have put the DNA replication in a culture which has N14, which is a light isotope, whereas the parental DNA contained N15, which was a heavy isotope of nitrogen. In the next generation, we see certain different observations. The information from the second generation let Messelson and Stahl determine which of the remaining models, semi-conservative or dispersive, was actually correct. So now now the concept was very clear that it is not the conservative model. It is either semi-conservative or dispersive model of DNA, which we are trying to understand. When second generation DNA was centrifuged, so after kind of 20 minutes, when it is again centrifuged, we see that the two bands were produced. One was in the same position as the intermediate band and the band of the first generation, and the second was higher. So one heavy strand was precipitated at the bottom, and intermediate strand was precipitated in the between, or at a little higher range than the lower one. This results two These results made Messelson and Stahl understand that DNA was being replicated in a semi-conservative way. It means hybrid was being formed. There was presence of even the higher or heavy metal because the parental condition was maintained, and even the presence of light one. The pattern of two distinct bands, one the position of the hybrid molecule and one at the position of the light molecule, is just what we expect for semi-conservative replication. In contrast, in dispersive replication, all the molecules should have bits of old and new DNA, making it possible to have a purely light molecule. So this is what they wanted to make everyone understand. You all can see conservative nature of DNA means whatever the parent is, the parental condition will be maintained. Here, a completely one. If this is from mother, this is from father, then the child will be having either the characters of mother or the characters of father. There will be no intermixing possible. But in semi-conservative nature of DNA, we see that it is not like that. Here, parental strand is definitely present everywhere. But whenever the complementary is being formed, that is the new strand. Whereas in dispersive mode of DNA replication, there will be few bands present showing that some of the DNA is from the parent, whereas some is from the new type. And this was not at all seen at any case, and first one was also not the case. So what came into light was the presence of semi-conservative nature, that 50% of parental hybrids were there, or parental type of DNA strand were there, 50 were new strands, and 50 were intermediate strands which contain both the parental as well as the new strand. And what do we mean by 50% parental as well as the new strand? We mean that this condition, this is a condition where N14 is also there, and there is presence of N15 as well. Suppose if I say this was N15 and the blue one is N14, so this contains both the things, isn't it? So that's why this is what Michelson and Stahl actually confirmed. Now, this is the idea by which they had done the entire centrifugation and separated the DNA. N14 is lighter, so it will make, or it will be present little upper than the N15, and N15 being heavier will be below it. In between will be the presence of intermediate strand containing N14 as well as N15. So I think it is little understood now. So this is the entire setup by which it was done. As the time proceeds, we feel that was zero. At first, we have taken a beaker. This is the beaker, and in that medium of N15, which is a, or N15 labeled N, which is nitrogen NH4Cl, we have taken and we have added the DNA for replication within it. Now, as the DNA replicates, it requires certain materials and definitely nitrogen. That nitrogen from NH4Cl will be provided for the formation of the replication, or for replication, and along with nitrogen, N15 isotope will also be going with the DNA strand. And when we try to grow it later, or when the experiment basically starts, we let this N15 label DNA be present in N14 medium. So now N15 will be the parental strand and N14 will be the new strand which will be formed. How new? Because all the nucleotides which will be added will be N14 labeled. So that is what we see. As the time proceeds, we can see orange color being present. So orange color represents that there is presence of semi-conservative nature of DNA. And with time, this will be completely understood that 50% will be light strand, 50% will be hybrid DNA. So with this idea, the Messelson and Stahl experiment was very successful, and they could conclude few things. In the semi-conservative model of DNA, each hybrid molecule from the second generation would be expected to give rise to hybrid molecule and a light molecule in the third generation, while each light DNA would only yield two more light molecules, because the environment is light, or the culture in which all the replication is taking place, it is a light culture. It has presence of N14. So now whatever will be formed, it will be containing N14. Parental strand will be just one, whereas the new strands, all the new strands which are getting formed, they all will be having N14 only, and that's why this is the case. Thus, over the third and fourth generation, we would expect the hybrid band to become progressively fainter, because it would represent a smaller fraction of DNA, and light band to become progressively stronger. Why? Because N14 medium is used, and only the presence of N14 will be detected. Even though parental strand will be present or hybrid strand will be there, but the concentration will be way too less as compared to the new strands which are getting formed. So as we can see in the figure, we have already seen the figure. So Messelson and Stahl saw this, thus just this pattern in their results, and confirm that semi-conservative nature of DNA is being followed. And what can we conclude by the end of this experiment? That semi-conservative nature of DNA is being, the nature of DNA is semi-conservative. It means half of it is parental, or one of the strand is always parental, whereas the new strand is formed which is complementary to the parental strand, and each of the strands of the DNA, they can act as a template.